It's a great pleasure to be making my first visit to Argentina. It's absolutely wonderful. So, as Herbert said, I'm going to talk about aging, and because it's an area of science that I suspect may be quite unfamiliar to some of you, I'll start with a rather general introduction and then come on to some detailed accounts of the work that we're actually doing in the lab. So aging is, of course, in the news a very great deal because of the increase in human life expectancy. It's a trend that started in the middle of the 19th century and has been going on at a more or less linear rate ever since, of about 2.5 years per decade. So I've just illustrated it here with the work of some demographers who've plotted life expectancy at birth against the year in which the birth took place for the country that was the world leader at the time. So to begin with, that tended to be the Scandinavian countries. Latterly, it's become particularly Japan. And the horizontal lines just show various people's predictions of when this trend was going to top out. But actually, there's no indication that it is going to top out, and it's not clear what the intrinsic limit on human lifespan is actually going to turn out to be. So, of course, this is something to be celebrated. People have a better health care, better lifestyle, they're staying healthier as they age, and so they're living longer. But it's coming with the huge downside that age is also the major risk factor for the predominant killer and chronic diseases, dementia, cardiovascular disease, and cancer. And this is therefore giving us a big scientific challenge. We need to understand the biological mechanisms of aging and then use that knowledge to try to intervene in the aging process itself to try and improve people's health as they age. But until recently, the prospects for doing that have not looked particularly rosy. And that's simply because aging is such a very complex process. If you look at what actually happens during aging in humans, numerous different forms of damage and pathology accumulate in different tissues, and it almost doesn't look as though there is a single underlying aging process. Rather, what you're seeing is independent processes going on in different parts of the body. And of course, if that were true, it would carry a very pessimistic message. It would mean that we were looking at best at piecemeal intervention into the aging process. And also, much along the same kind of argument, I think people have tended to assume that our model organisms, about which we've already heard quite a bit, will age very differently from each other. They have very different diets, lifestyle, metabolism. The mouse is warm-blooded. Why should they encounter the same kinds of damage as they go through their lifespan? And of course, if that were true, we wouldn't be able to use the usual biomedical agenda of using the simpler organisms to understand the more complicated ones. But fortunately, this pessimistic view has been overturned in recent years. It turns out that aging is malleable, so single gene mutations can extend lifespan, and some of the first ones that were discovered were actually in Caenorhabditis elegans. And they turned out to be in genes that encoded an invertebrate insulin-like growth factor signaling pathway. So when activity of the pathway is reduced, the worm lives longer, and also very importantly, the mutant long-lived worms are still wriggling around and active when all the controls are dead. It's not just an extension of the moribund period at the end of the life of the worm. And perhaps almost as surprisingly, it turns out that at least some mechanisms of aging must be conserved during evolution, because mutations in this same pathway can extend lifespan in Drosophila. This is an example from our own lab. This is a null mutation in a gene called Chico, which encodes the single fly insulin receptor substrate. You can see you can get quite a good extension of lifespan if you remove one or both copies of this gene in the fly. And also there's conservation to the mouse. So this is, again, a collaborative venture that I was involved with Dominic Wither's lab in London. And these are data for a Chico mouse. So this is a mouse that's lost both copies of its insulin receptor substrate 1. You can see the increase in lifespan. One of the interesting things about these mutations in flies and mice is they extend lifespan mainly in females. The effect is much stronger in females than in males. We don't yet understand why that is. But very importantly in this study, they looked really rather carefully at the mice as they went through middle and old age to look at their health status. And what they found was that they preserved youthful levels of physiological function on quite diverse things. So glucose homeostasis, immune profile, and motor function. And they also showed a delayed onset of specific aging-related pathologies, so osteoporosis, cataracts, and ulcerative dermatitis. So this is what we were not expecting to see, a really very broad spectrum effect of a single mutation in a single gene on the health of the animal as it goes through the aging process. It's very early days to say what's going on in humans, and the main approach that's being used to look at whether these pathways are relevant to human aging is genetic association studies. So people have simply taken the orthologs of candidate genes from the animal studies and looked at whether genetic variants near them associate with human survival to advanced ages, so 90 or 100. And quite a few of them have come up with positive results for the IGF pathway and also for one of the key fork head transcription factor effectors of insulin and IGF signaling. I just included this lady on the bottom right. I hope you can all see her. Centenarians are a very interesting bunch of people. They definitely don't get to be 100 because of their healthy lifestyle. They're very average in their general habits. Now, of course, what we're talking about here is a nutrient sensing network. It detects the nutritional status of the animals and matches their metabolism, their growth, their reproduction, other energy consuming processes to their nutrient status. And it's been known for a long time that reduced dietary intake itself can increase lifespan. It was first discovered in rodents in the 1930s and has since been shown to apply in a very wide diversity of organisms. And importantly, I think, for the applicability of this whole story to humans, a paper was published in 2009 showing that dietary restriction extends lifespan in rhesus monkeys, and it also improves their health during aging. I think we'll probably never know whether the dietary restriction increases lifespan in humans because we can't do a randomized controlled trial. There would be no compliance. It's a very difficult thing to do. But some people do voluntarily dietarily restrict themselves. And most of the health indices that they show are very similar to the ones that are seen in dietarily restricted rhesus monkeys and mice. So I think what we have growing here is a proof of principle that we can actually target the underlying aging process to protect against aging-related disease, at least in model organisms. So that's the agenda, really, in this area of research. So what I'd like to do is, for the rest of the talk, is briefly to address just two studies that we're pursuing in the lab. One is to do with dietary restriction, and then I'll come on to some work that we're doing on neurodegeneration. So we've had a wonderful introduction to Drosophila and Drosophila diet from Bill Hansen. And the way that we usually restrict how much the fly eats is simply to dilute that food medium that you saw. It's a mixture of yeast and sugar, generally. And if you dilute the food, female fecundity goes down with food concentration, lifespan goes up, and then falls off a cliff through starvation. And this is a very typical response to reduced food intake in all of the model organisms. We know that this is not just a response to the calories in the diet. So sucrose and yeast have about the same caloric content as each other per unit weight. And if you do this dilution trick, either with the sugar or with the yeast, the flies barely respond to yeast dilution, and almost all of the action is in the yeast. So we're certainly looking at an effect of specific nutrients rather than just overall calorie intake. The other thing to say about this diagram is that there's been a general assumption, I think, that this reduction in fecundity and increase in lifespan are somehow causally connected with each other, that the flies have to reduce their reproduction in order to live longer under dietary restriction. So what we've been doing with this is trying to look at the nutritional basis of these responses, looking at the effects of the different components that are present in the yeast. So what the two people who've done this work are Richard Grandison, who was a graduate student in the lab, and Matt Piper, who's now actually running his own lab in London. And what they did was to simply take flies that were dietarily restricted and add back to them those components that had been measured as present in the yeast that we were using in our experiments. And the first thing that turned up was that, as far as fecundity goes, all of the action is in the amino acid portion of the yeast. So we're certainly looking at an effect of specific nutrients rather than just overall calorie intake. The other thing to say about this diagram is that there's been a general assumption, I think, that this reduction in fecundity and increase in lifespan are somehow causally connected with each other, that the flies have to reduce their reproduction in order to live longer under dietary restriction. So what we've been doing with this is trying to look at the nutritional basis of these responses, looking at the effects of the different components that are present in the yeast. So So we've got here on the left dietary restricted flies, on the right fully fed ones. You can see the difference in fecundity. If you add back any of the components of the diet apart from the amino acids, fecundity doesn't respond, it stays low. Whereas if you add back the amino acids, you get the full response. The same thing turns out to be true for lifespan. So if we look at the left now, the long-lived flies are the dietarily restricted ones. If we add back everything except the amino acids, their lifespan stays long. But if we add back the amino acids on the right, down it goes to fully fed levels. So this is, of course, consistent with the idea that somehow reproduction and survival are trading off against each other through reallocation of these amino acids. But when we start to look at which amino acids, that association actually falls apart. So it turns out that the response of fecundity is entirely dependent on methionine. So if we give them back methionine to the level which it's present in the fully fed yeast, we get as big a response in fecundity as if we give them all of the essential amino acids, or if we fully feed them. So methionine is entirely accounting for this effect on fecundity. However, if we look at lifespan, if we give them back methionine, nothing happens. They stay at the long dietarily restricted lifespan. So we can pull these two traits apart simply by rebalancing the amino acids in the diet to get a long-lived, fully fecund fly. And it turns out that a mixture of the essentials is what's important for reducing lifespan. And what we think is going on here is that there's an imbalance in the essential amino acids in the diet in comparison with the ratio in which the fly wants to use them. And that's what's causing the shortening of lifespan when we fully feed the flies. Finally, it turns out that this insulin signaling pathway that I've mentioned to you is very important in mediating these responses to dietary restriction. And that's shown here with the responses of a control wild-type fly and of a mutant in the fly insulin receptor. It just has one insulin IGF receptor, and these experimental flies are carrying a dominant negative mutant version of it. So the story for fecundity is in the blue histograms, and you can see that in the wild-type flies, if we add back methionine, essential amino acids, or fully feed them, we get a nice increase in fecundity. If we do that to the insulin mutant flies in the darker blue, nothing happens. They're completely impervious to these additions of diet. Similarly for lifespan, with the red lines at the top, if we look at the controls, it takes all of the essential amino acids to bring down lifespan or full feeding. They don't respond to methionine at all. The insulin mutant flies are very long-lived, and their lifespan doesn't come down at all in response to the manipulations of diet that shorten the lifespan in the controls. To summarize this first experimental bit, the crucial thing in the diets for these responses are the amino acids, not calories. And it's an imbalance in the essential amino acids in the diet that's crucial, and insulin signaling's mediating the responses. And one of the ways that we're taking this forward at the moment is to look at whether the same is true for the effects of amino acid balance in the diet in mice when they're dietarily restricted. Second thing I'd like to say a little bit about is neurodegeneration in flies. We know that this pathway can extend lifespan, and it seems to be an evolutionarily conserved effect. But of course, what we're interested is in function during aging. Are the animals staying functional? And if so, what exactly are the molecular mechanisms by which the pathway is achieving this very broad spectrum effect? And we're probably only going to find that out by understanding what's happening in each system. So we've been concentrating on the nervous system in the fly. And the way that we've been looking at functional decline in aging is to look at the giant fiber pathway, which is the neural circuit that does the escape responses of the fly to a visual stimulus. So if it sees a frightening stimulus, it jumps and then it flies. And the giant fiber system is the pathway that mediates it. It consists of two very large interneurons, which then synapse either directly with motor neurons or with other interneurons, and then the motor neurons that activate the jump and the flight muscles. And we can make electrophysiological recordings from this system by stimulating at the front end in the brain, and we can measure the length of time it takes for the signal to get to the muscles, and we can also look at how good the whole system is at following a high-frequency train of pulses. So the person who's been doing this work, which is unpublished at the moment, is Herve Augustin, who's a postdoc in the lab, and he's trained, he has a neurophysiological background. And the first thing that he showed was that the speed of conduction through this system to the two sets of muscles goes down with age. In other words, the time that it takes the stimulus to get through increases. So the system shows an age-related decline in its function. And he also showed that if he made the flies constitutively and systemically mutant for insulin signaling, that he could protect against this decline. So these are these same flies that you've already seen that are expressing the dominant negative version of the insulin receptor throughout life and throughout the body. And we've got the two sets of genetic controls for this on the left. This is the fly GAL4-UAS system for misexpression of genes. And in the controls, we see the age-related decline in function again. It's blocked in the mutant. So something about lowered insulin signaling in these flies is preventing the age-related decline in the function of this system. What we would like to do is to bypass the developmental effects of these mutants, because one of the things that the pathway does is to make the flies reduced in size, which complicates things if there's a mutant in the insulin signaling system. And also, we'd like, within the adult itself, to be able to lower insulin signaling in specific tissues to ask where this restorative effect is coming. And the way that we do that is using these inducible drivers. So the GAL4-UAS system, as many of you probably know, has been modified so that the GAL4 only activates the upstream activating sequence in the presence of an inducer in the food, mifepristone, RU486. So what you can do is to make your flies, but in the absence of the inducer, there's no gene misexpression. But if you add RU to the food, then you get the misexpression coming on. So this allows you to do both timed and tissue-specific misexpression of the genes. And what Augie has done here is to bring the flies through development normally, and then he misexpresses the insulin receptor dominant negative just in the neurons of the fly. And he finds that if we do that, we have a slightly different setup here, because what we do is compare induced and uninduced rather than driver and driven. What he finds is that whereas in the uninduced flies he sees the age-related decline, it's abolished if he drives down insulin signaling in the neurons of the adult fly. So this is a nervous system specific effect. In fact, he can pin it down further than that. There happens to be a neuronal driver whose expression pattern you can see on the right here that only drives in the giant fiber system. And if he just drives that, then he rescues the age-related decline. So it's a real system-specific effect. So he started to get into the mechanisms of what's going on here. And there are two types of synapses in this system, electrical and chemical. And he's looked both in terms of gene expression at the chemical synapses, that's shown here. He's also looked functionally because they're localized in particular bits of the circuit, so he can isolate their neurophysiological function with age. And they actually don't show much decline with age, and it's unaffected in the insulin mutants. So it looks as though the chemical synapses are not part of this insulin rescue story. But the electrical synapses are. So here, we're talking about gap junctions, which are made from a protein called shaking B in Drosophila. And what Augie has done here is to stain in control and mutant flies at two different ages. And you can see in the young flies on the left that the basic pattern of staining is that there's, at the front end of the fly, actually in the thorax, there's medial staining, and then there are these two lateral tracts which are going out towards the muscles. And during aging, in the control flies, the staining in the lateral tracts gets